my name is Frank Amort. I'm EATG's Combination Prevention Chair, and I'm moderating today this session. There will be a presentation by Simon Collins, who is one of the co-founder of HIV iBase, and he edits the HIV treatment bulletin and is well known in treatment information services and community engagement with medical research. The HIV iBase is a London-based treatment activist group, and today's webinar is going to give an update on recent developments on COVID-19 vaccines. It's actually a topic where every day you get new information and news. Maybe the most recent news we got from many European countries that EMA is the European Medical Agency is going to publish their statement earlier this year, and testing also within the European Union might already start within this year. But having said that, I would kindly ask Simon to give his presentation for today.
So thanks, Frank, for that introduction. I'm a bit concerned we might have an echo or feedback because I'm using these two screens. So if that happens, I'll just have to log out of one and then share the screen on my laptop, if that's okay.
It's fine at the moment.
Fantastic. So this is the top bit is really important. This is just a non-technical overview of the research and developments over COVID vaccines over the last few months. And we're also really lucky to have Richard Jeffries from TAG, who can answer much more technical questions than I can. So the slides are very simple, but we've got facility to answer all sorts of geeky stuff too. So the background really is that we've known right from the start of the pandemic that effective vaccines will be the only way of returning to some sort of normal life. And so we're going to talk about what's happened, mainly three different types of vaccines. There's a huge range of current candidates, so I'll give some information about that. A little bit of summary information from the results from the three leading vaccines, and then the whole issue of access and fair global distribution. And as a caution, I'm not an expert in any of this. This is just the information I've picked up just from mainly reporting treatment. Right, I've got a delay. I'm clicking one slide. It takes a while to come through. So the real basics, so that everyone's starting from the same, the same basic understanding. Really, is that the vaccines trick your body to produce a strong immune response. And we do produce immune responses naturally to all sorts of organisms like HIV and like coronavirus, but we don't produce them quickly enough in order to control the really serious infections. And so a vaccine will sort of prime your immune system so that then if you come into contact, your immune system is already ready to start fighting straight away. Although early vaccines often used the actual organism or a weaker dose of the organism, modern approaches are much safer. They only use a little bit of an inactivated virus, for example, or in some of the newer approaches are getting your body to make these proteins that look like the virus. So I needed to put in a picture slide. This is from WHO earlier in the year, and it's a sort of graphic of the different ways that the main candidate vaccines work. These are the ones we're going to talk about today. Messenger RNA is probably the leading vaccine candidate right now, and it's important because this is the first time we've had this sort of vaccine. delay. We are. So messenger RNA, it's a bit of nuclear material that's related to the virus. And although it's been in research for a long time, none of these types of vaccines have been approved yet. And if you think back to the way that vaccines were being talked about in March and April, lots of experts were using this as a caution, saying there's never been an effective mRNA vaccine before. And it's just fairly ironic that the two leading compounds that have come through both use this approach. They work by injecting directly into, in this case, muscle cells, some of this genetic material, and then the muscle cells themselves produce a version of the same protein that's on the surface of the coronavirus. It produces then the immune system produces a response against these proteins, and then I had a really good example the other day. The mRNA itself is then quickly broken down, and I haven't used snapshot, but it's a bit like that. You put in this genetic material, and then it just gets broken down quickly, like the photographs that disappear. Apparently, it can produce easily in a lab and easily scaled up. The second version is viral vectors, which is where you still use you're delivering to your immune system a little bit of the surface of the virus that you want to generate an immune response to, but here you load it onto an inactive virus. So there's two parts of these vaccines. There's a harmless virus, and then inside that harmless virus, you've got some of the proteins that are related to the coronavirus. The Oxford vaccine uses this approach, and so also CanSino and Janssen. They all use different adenoviruses though. So the Oxford virus is using a chimp adenovirus. They can produce both sorts of cellular responses, T cells and antibodies, and then there's some cautions about this, which is maybe if you've been exposed to this version of the adenovirus before, it maybe limits the number of times you can use it, or you can have booster vaccines. So you might need to start off with this vaccine and then use a different vaccine to have a booster dose. There are two main types, replicating and non-replicating viral vectors. Non-replicating just produce the surface protein, and replicating also would produce the carrying virus as well. Missed the slide. Protein-based. So protein-based is sort of the third type of virus that it's useful to know about. It's being used by Novavax, and also a GSK version that's coming along later. And the difference here is you use, there's two parts again to the vaccine, and here you use something called an adjuvant in order to prime your immune system to respond better to the surface proteins. Much of this work started really early in January 2020. So before this was on our radar, by a couple of months maybe, as something that was going to be as serious as it turned out to be, vaccine groups, including some of the commercial companies, were working on either directly on SARS-CoV-2, or they were previously working on Ebola or MERS vaccine, and then they transferred all that research to the new coronavirus. The pipeline includes hundreds of potential candidates, and over 40 of these are now in human studies. And although we have amazing short-term efficacy, far better than the FDA suggested would be good. The FDA was hoping for 50% efficacy to approve a vaccine, and now we're talking into the 90s in terms of efficacy. These vaccine studies are not based on HIV transmission. So nearly everyone assumes that we're looking at the vaccine in order to prevent transmission, but those aren't the endpoints in the studies. The endpoints are whether or not you have symptoms of coronavirus, and then those people with symptoms are tested to find out if they're positive or not. The Oxford vaccine does have transmission endpoints. And the big picture is really, in order for this to be effective, we need universal global access. The economic outcomes of all this are really unclear though. So this is a graphic that just shows the range of candidates. There's over 300 candidates, more than 40 are in human studies, and of these 12 are in phase 2-3 studies. The next slide shows the same compounds by region and funding. Here we go. And you can see that the bulk of the research is being done in high income countries. And although a good percentage of it includes public funding and academic research, there's also a large input from industry. This is a summary of the human studies. These are the ones in phase 2 and 3, and the ways they work really. And the next slide is actually much more complicated, but the slides will all go online. So I wanted to include this as a reference slide. It's from about a month ago now, so some of these compounds might have shifted along the development phase a little bit more. This was a very nice slide from Barney Graham from an AVAC talk earlier in the year from a community perspective. And you can also, not sure how they show on your screens, but they've got little flags there to show the countries that are developing these, and also the type of virus and the stage of activity. So this is a very useful reference slide to use afterwards. The red box there shows, I'm not sure I should go back. The red box there shows which candidate vaccines are being, up here, shows which candidate vaccines are being used by the COVAX funded program. And COVAX is the global initiative by WHO and other organizations in order to enable global access by low and middle income countries as well. The results for each of the main compounds are worth knowing now because because some of these are actually very, very recent. The latest data has only been available on the Pfizer compound for about a week because the FDA does an amazing job of putting all the documentary evidence online so that there's open access to see all the details from these studies. The top line results from the main phase 3 study in about 43,000 adults was released about a month ago, and then now we have the full results showing about 95% efficacy at preventing serious COVID infections, or sorry, preventing symptoms really. And so you had 162 cases of COVID symptoms in the placebo arm versus 8 cases in the active drug arm. And then the next stage is who became hospitalized and who had really serious infections. And so the severe cases occurred 9 in the placebo group and 1 in the active vaccine arm. This vaccine's already been conditionally approved in the UK and on the 2nd of December, and then in the US just over a week later. And then the interesting thing about development by Pfizer is that they chose not to accept federal funding to help the development. So this is now a private initiative, and everything that goes along with that means that the companies really can decide what they want to do in terms of marketing and sales. The other practical point is it requires something called ultra cold chain. It has to be stored at -80 degrees, and that's a challenge even for many high-income countries. It's very difficult to guarantee this in transport, and so the practical issues are going to be a problem. Also, I've got to mention here that 96% of the available and the planned supplies of these vaccines have already been allocated to high-income countries. The next slide is for the Moderna vaccine. And again, both these studies started on the same day, actually. I think about 29th of July, and they both presented results sort of on the same day, and they've both simultaneously gone to the FDA for review. The study here was a slightly smaller study, 30,000 adults, all in US-based sites. Very similar efficacy. The interim data was 90 versus 5 cases, 90 in the placebo group. You will hear this first because this document only went online yesterday in terms of the FDA hearing, but the full results so that there were 185 symptomatic cases in the placebo group compared to 11 cases in the active arm. And again, what we really want to know is the severe infections, in this case, were all in the placebo group. So all the people that got hospitalized with coronavirus and COVID-19 were in the placebo arm of this study. The FDA hearing's in a couple of days. I don't think there's any way it won't be approved. It's slightly easier to transport. It requires cold chain, but only down to minus 20 degrees. And then 100% of the upcoming manufacturing capacity has all been committed to high-income countries. And then the third results slide, they're not as final as the previous two, is for the Oxford vaccine. It's had lots of publicity. This was a program that was definitely working on other viruses, and they transferred this to coronavirus. The top line results are that efficacy is 70% overall, but there was a sort of prescribing error, quite a serious one really, in that UK participants were all given, it's 2-dose vaccine, like all the vaccines so far. It's a 2-dose vaccine, but all the UK participants were given a half dose for their first vaccine. And this lucky accident really resulted in much higher efficacy. So 90% protection if you first dose uses a half dose, compared to 62% protection if you were using 2 full doses. The Brazilian sites have got much better ethnicity breakdown. I think maybe 65% of Caucasian in Brazil and 90% in the UK or something. This is really important issue in all studies, really. The other outcome from the dosing error was that rather than getting the second dose 4 weeks afterwards, I haven't really heard a discussion about this. Rather than getting the second dose 4 weeks afterwards, it was separated by 12 weeks in at least half the UK participants. And I'm not sure, I'd be interested to know if Richard thinks this makes a difference to the response. I think there was some US funding. There's an ongoing phase 3 study using the half dose in the US, which will report in a couple of months, and this might be critical to getting approval. Importantly, there's no cold chain involved, and also importantly, they are making this available at a very low, $4 a shot approximately, and that 64% of this is pledged to low-income countries, although from what I can tell, most of that's to India. The preliminary data looks amazingly exciting, way better than anything anybody would have predicted at the beginning of the year. We need to know the duration of protection. We need to know whether there's a booster, how long until you need a booster. We need long-term data on safety and head-to-head studies, maybe comparing the different vaccines to each other. We'll need further data on subgroups by age. There's a question about using adenoviruses. There was an HIV study many years ago, about 12 years ago, called STEP, and it didn't work as an HIV vaccine, but there was a slightly higher risk in some participants, male participants who are uncircumcised, acquiring HIV. So this is a safety question we need to monitor. CanSino are already looking at this. We need to know how to safely use the vaccine based on levels of other healthcare issues. So far, we're not expecting anything difficult with HIV, but also other inflammatory conditions, including allergies and lupus and a whole range of complications that sometimes are difficult with vaccines. We need pregnancy data, breastfeeding data in children, and we need to know how it works if you've already had coronavirus. There's been some concerns about the speed at which vaccines have been developed, and I'd like to sort of say that nothing has been, none of these steps, these are traditional steps, have been stopped. They've really just been compressed so that one stage starts while the other stage is running, including this earlier scientific evaluation. So in Europe, there's a rolling evaluation where the data as it's produced is going to the regulatory agencies, and it makes it easier to make a decision afterwards. Also, scale up started much earlier, before we knew whether or not the vaccines would be so effective. I've put the link in here. I've got about a long pause between slides. I'm changing slides on one computer. So there's a link to these protocols that are all online. They're not technically all online because they get updated and revised, and some of these links don't include the latest updates. And I also included the Janssen vaccine, which is about to start. So the Janssen vaccine's got a window where the current vaccines haven't really been broadly accessed yet, so it's still ethical to use a control, a placebo control, and that's about to start. And I'm not sure, I think this might only be a single dose vaccine. I'm not sure, that really needs to be checked though, but that would make a big difference. Some of the vaccines in development only need one dose. So then the big problem is, or the big issue is vaccine access, and without global access, none of these benefits will help us as a planet, really. And there was a very good focus on access. The last couple of days, it was organised by the African CDC, and there was a lot of talk there about vaccine nationalism and how high-income countries have bought the first stocks plus options for the next stops. And so the access plans, even though the WHO and international organisations have been working on plans to ensure equitable access across the planet, this is based on much lower coverage. And even the best case at the moment is for 20% coverage is still too low for herd immunity. My next slide has disappeared. Unfortunately, it showed very nicely how high-income countries had purchased much more than COVID. These slides, which I asked the EATG to check, but that's my problem. I'll try and come back to these at the end when we're in the Q&A session. They were produced. I have to move on here. So they're graphics actually, and they really just show the relationship between the pre-bought and reserved stock of a range of countries compared to their populations. And it shows that most countries haven't been able to secure enough vaccine for 100% of their population. And then some countries were in positions to buy more. So for example, the UK has bought or has options on 3 or 4 times the number of vaccines that it needs to treat the whole population of the UK. Canada has done this by about 6 times, and the US has done it as well, I think, by about 2 or 3 times. So some countries have got huge reserves of vaccine supply, and the hope is that these would immediately be turned over to international programs. These are the organisations that are leading on global access. So the CEPI was set up, the Coalition for Epidemic Preparedness was set up a couple of years ago in order to be, luckily had the foresight to realise this might be a problem. And then the COVAX is a vaccine arm of initiative set up by the WHO to generate best responses both to treatment and to vaccines. And it's co-sponsored by the Global for Gavi, which is Global Access for Vaccine Initiative. People's Vaccine Alliance is a coalition of NGOs mainly that are also interested in equitable access. This is my only slide from Anthony Fauci, who's given lots of lectures, but it highlights this issue about whether when the vaccines are available, people will be keen to use them. And lots of discussion groups on HIV forums show that there's one set of people who are really queuing up to get the vaccines as fast as possible to return to normal life, and there's still a level of uncertainty amongst many about just many people who really, for some reason, are uncertain in spite of all this global crisis. You know, the huge number of people that have already died and become infected. People are still curious about whether the vaccines are a good thing, and we need to overcome this. And then the second slide was from the African Forum yesterday, which really just in more detail breaks down this issue about denialism and hesitancy, and also highlights that some people are doing this actively trying to dissuade people from using the virus, from using the vaccines. And so this will be a key role for community to be involved in explaining and education. For UK access, just to give feedback on how things are working here, the National Health will provide the vaccine free to all people living in the UK. There are 9 priority areas. The highest risk rightly gets first access by the oldest people if they're living in care homes and frontline healthcare workers. Severe health conditions that limit people from leaving their house is in group 4, and other complications, big list of other health complications is in group 6. So HIV is included as a priority. And there's some very good guidelines produced by BEVA, which are now online, that also have got a bit of information about safety and other concerns. So just the last couple of slides are just some of the questions that came up from members of the UK CAB for a training that we have planned, and you can see that they cover really reasonable range of just general questions: whether vaccines are safe, whether there's a risk of catching COVID-19 from the vaccine, whether there's side effects from similar to the flu jab, whether it's good to take it now or to wait, who's going to give the vaccine, is there a possibility of interactions, and hopefully we'll have some of these questions we can talk about today. Lots of questions focused on whether the research had been carried out in all sorts of different groups of people, whether their ethnicity had been considered, whether ethnicity had been considered when the vaccines were approved, the interests of different communities being considered, whether black people were treated like white people in terms of the way the studies were run, if there are interactions with chems, crystal meth, or if someone's had hepatitis C. Lots of personal questions that really we need to produce a good set of community documents that as advocates and activists we can help. Now we have really the tools that we wanted in terms of effective vaccines that are going to be available soon within the next year. Hopefully for most people, within the next few months for most probably high-income countries, and within the next year for low and middle-income countries. All those slides were really about access. I'm going to go back and try and see if they show properly on a PDF version while we start the chat and the conversation, in case I can show those later. So apart from that, I'd like to just see how we're doing for time. So we're just coming up to about 20 minutes, which is good. So we've had unparalleled scientific advances, really, global access now, at best, is planning to reach 2 million people in low-income countries over the next year, maybe over the next 2 years. So this isn't an instant solution for most people who are in need of the vaccine. And we need long-term follow-up. And other than that, I'd like to thank a few people for slides, none of whom I was able to check first, but I think in the interests of community use, these are all okay. And then the most interesting part now will be the discussion where we all get to talk to each other.
Okay, thank you very much. You have mentioned it, and I've forgotten to say it before, that with Richard Jeffries from the Treatment Action Group in New York also is joining today's webinar, and he might also answer some of the questions. And there is one question in the chat from Ben Cromarty, sorry for not spelling the name maybe correctly. Is there any explanation for why the half dose data is better? So this whole information around that in one of the study arms, there were better results with people receiving only half dose.
So I'd probably ask Richard if he'd like to comment on that.
Yeah, unfortunately, I think people only have guesses at the moment. The one that I've seen sort of most frequently is the idea that perhaps because the vaccine is using this adenovirus, the harmless virus, as a sort of carrier for the coronavirus protein, that perhaps the first low dose, it doesn't trigger the immune response against the carrier adenovirus so much. So when they give a second higher dose, the immune response isn't blocking the adenovirus from getting in and delivering the coronavirus protein. So it's sort of like the lower dose sneaks it under the radar, and then the second dose is able to work better because the immune system isn't rejecting the adenovirus carrier. But it's just a guess, and I think that's going to be looked at in a lot more detail.
Okay.
So what would have happened if this hadn't have happened and we had 62% efficacy with the Oxford dose? Would that still have gone ahead, do you think?
I think it might not have, because at least in the US, the FDA, in addition to requiring an efficacy of over 50%, they had some sort of a spectrum of what they call the confidence interval around that result that needed to be above a certain threshold. And I'm not sure if that would have held for that particular result. I think the bottom sort of estimate needed to be above 30%.
Okay, so it's quite a lucky accident then. This is a very serious response. Imagine only giving half a dose of a treatment. So,
yeah, I know it was some difficulty associated with trying to measure how many adenovirus particles you have in a vial. It's not as easy as you might hope, apparently.
Okay, thanks.
I have, there was one comment that the Janssen vaccine is only one dose. Maybe you can give an overview which of the different options have one dose or more doses. Maybe that's for some people.
Yeah, I think the Janssen vaccine is the only one I know of that's giving a single dose, certainly in an efficacy trial at the moment. That there's usually just two doses, what they call a prime and then a booster.
Okay. And there is a question from Brian Teixeira. Given that there are, and there will be various vaccines with high efficiency, is there assumption that there will be, so we can assume there is a choice. What is the benefit? So I would understand at the moment, I start thinking about this response. We have the Pfizer biotech approach, which is very complicated from the logistics, while the Oxford Astra is for the health system way easier to handle and to organize. I understand also that it's cheaper. But what would you say? I also understand that the Oxford Astra already has some studies and information available about the prevention of new infections, while Pfizer Biotech only about the seriousness and the complications. So at the moment, it's a little bit, you get what is available. And at the moment, that's Pfizer Biotech. But let's imagine in half a year or a year, if we might have 3, 4, 5 vaccinations, what would you think then would be the choice to take from?
So I think it's complicated. You have to work with the best information we have at the moment. And the fact that there's going to be a delay on access, no country is going to have universal coverage because there's a limited supply. And so as the people who are most in need get first access, there will be additional safety data that get collected as we go along, and research in other vaccines is also going to continue. So the pace of research is incredibly fast and changing, and if you get a good response from one vaccine, it doesn't really matter which vaccine it is. You know, although there's these small differences between the vaccines, all you're interested in is whether you get a good response. And if it turns out that some of the earlier vaccines, for example, don't last so long, then there'll be other options for boosting later.
There is one question from Aizulu, and I think, well, I read it, and I think for some point she's already giving the answer, but maybe you and Richard can comment on that. What she's commenting is that so far, the clinical trials don't include specifically the person with suppressed immune system. So we don't have any data and evidence on cancer patients or person living with HIV. Are you aware that such data or trials are in preparation? When might we have such data?
Is that one you want to do, Richard?
Sure. So there was about 180 people with HIV in both the Pfizer and the Moderna efficacy trials. So not a huge number, and they were split between the vaccine and placebo group. So the Moderna was about 90 in vaccine and 86 in placebo, or somewhere around that. So they have a little bit of safety data in people with HIV, and the reports don't mention any issues arising, but there's not enough numbers to do a sort of analysis of efficacy just for people with HIV. But because people on treatment have, you know, comparable usually immune responses to routine vaccines like the flu, etc. I think the hope is that these vaccines will work just fine. It may be not more of an issue if someone is not on treatment and has uncontrolled HIV viral load or low CD4 counts. It could be that the immune response isn't quite as good, but I think for most people, the expectation is that they will work just as well as in an HIV negative person.
And actually, the approach from all the research, from all these studies, is you start off enrolling people who are least complicated and most likely to show a clean vaccine response, and then steadily, so long as it continues to be safe without any signals of concern, then slowly they enrolled older people, extending it to above 60, then above 70, then above 80, and they'll do the same thing in terms of other infections. They might, the more careful you have to be because of other health conditions, you might get the vaccine at a centre where you can be monitored afterwards. So because of the anaphylactic reaction to 2 healthcare workers, the Pfizer vaccine is now being recommended in the UK to only be given in settings where there's support in case you have a reaction. It's certainly if you have a history of anaphylactic reactions, not to use the Pfizer vaccine so far. Frank, are you able to share my slides again? So instead of the window that I'm looking at now, you can show the window which is the PDF version.
I can change that, but maybe the office can change it. So I think you are sharing at the moment. So
I'm still sharing, and I've got a PDF version up that's, if we get questions about access, which would be better than looking at this slide in the background.
Okay. There is one question from Ramon Espacio from Spain. I tried to summarize it. You said that in the UK, persons living with HIV have been included in the priority vulnerable groups.
Yep.
And they have in Spain, they have a discussion on that. And I understand that the Spanish National AIDS Plan is arguing that the HIV patients are somehow a younger population, well-controlled population, and so on. And there might not be a necessity to include them in the vulnerable populations. What was the argument in the UK, and do you have other experiences if person living with HIV should be included or not in the priority groups?
So great question. I can't provide. Well, great question actually, because the data on risk for HIV positive people from coronavirus started out being very optimistic. That in very small studies, you couldn't really see either an increased risk of infection or an increased risk of more serious COVID-19. As those studies progressed during the year and they became larger, including certainly there's a large study from South Africa, there's a large study from the UK which called the ISARIC, linked up with ISARIC databases, and these very large studies found an unexplained higher risk for HIV positive people, where we were more likely to be hospitalized at much younger ages. So HIV positive people who are hospitalized with COVID-19 could be in their 50s, commonly in their 50s or 60s more often, compared to people in the general population who would be in their 70s or 80s. So there is a difference there, and it's not explained by the factors that are related to worse COVID outcomes. It's not explained by background comorbidities or any of those factors. So I'd use that data. I'd knock that data back to the committees and the groups in Spain, and that sort of is enough to justify HIV positive people or HIV as an existing condition being included with other comorbidities.
Yeah, I think that's maybe a very important point to follow up on a European level, how the different national agencies are handling that, which populations they're including or not including, and what the rationales are behind there. There is one question, maybe I forward it to Richard. If you would see Pfizer in comparison to the others, is there a kind of superiority in comparison to other vaccines, or is that a similar effect? That's a question for Maka.
Sure, I would say that the Pfizer, BioNTech, and the Moderna vaccines look really very similar. From the information so far, both of those, the Pfizer and the Moderna vaccines, look slightly better than the AstraZeneca vaccine.
Okay. And then there is a question from Ben. It's referring to the phase 3 in trials, and I would like maybe to ask you, before you answer the question in detail, that you explain what the phase 3 trials are about. He's arguing that they might be considered ethically difficult because there are, what he's saying, effective vaccines, and comparing it with a placebo arm might be ethically difficult. But I would say it has also to do with the different outcomes, and still this phase 3 data might be needed. What would you argue, Simon and Richard?
So actually, that's a great question. Please, could someone from the office share my screen again? Fiona, maybe.
I think. Can you put the PDF up on the same computer that you have?
So I have the PDF is up there.
Please, can you close the PowerPoint?
So the only thing that shows is okay, so I'll fiddle around with it. So the phase 3 studies, are big, involve tens of thousands of people, and they're the real life data to find out whether the vaccines work or not. And you're absolutely right that all through this year, it's been easy to have placebo controlled. So half the people are randomized to get the active vaccine and half get a placebo, which is not active at all. And then you've got a very clear outcome about whether the vaccine works. As soon as vaccines become widely available, they're not quite widely available yet, then it does bring up ethical issues about continuing to use a placebo control, because if someone in the general population has got access to the vaccine from their public health system, why would they enter a study where they have a risk of getting the placebo? And more importantly, the people who you really want to know whether the vaccine works or not will definitely be getting access to the active arm. So the older people will be prioritized and comorbidities will be prioritized, and then you need to be shifting to an active control arm where maybe the control is the Pfizer vaccine or the Moderna vaccine. So that then you've got head-to-head studies, and they become a source of other information, which is important to shift that sort of design. There's also ethical questions about what you do now for people who are currently on the studies who received placebo in the current studies, say for Pfizer and for Moderna, and some people say that they should immediately roll over to be able to get access to the active arm, and other people are sort of suggesting that they should only get access to the active vaccine when it becomes available for people in that risk in their country. Okay. And there's reasons to support both. Probably the last of those options, you only get access when it would become available for you, is better, because you then carry on the placebo control data will be a bit more meaningful for a bit longer.
Mm-hmm. There is one question about referring if the mRNA approach, which is used in the Pfizer-BioNTech vaccination, if that approach might have implications for the development and search of HIV vaccinations. Maybe Richard?
Sure. So I think there are some mRNA vaccines for HIV that haven't been tested in clinical trials yet. There has been a therapeutic mRNA vaccine for HIV that's been tested in the Netherlands, I think it was. And I think there might still be some ongoing work related to that. I think the big challenge with HIV that's different from coronavirus is that with coronavirus, that we know that this spike protein can induce antibodies that are very effective at blocking the replication of the virus. And so that was an obvious choice to put in a vaccine. And thankfully, it worked. The antibodies that and immune responses that were induced by that spike protein were protective against disease. The big issue with HIV is that we don't know, there isn't a component of HIV that can induce immune responses that are protective because of how well the virus has evolved to avoid antibodies. It's very difficult to find a part of the virus that will induce an antibody capable of inhibiting a wide range of HIVs, and so that's still being worked on. And so the delivery platform like mRNA or an adenovirus is not so much the issue for HIV. It's which part of the virus you need to put in the vaccine to trigger an immune response that will protect people, and that's the part that really needs to be figured out.
Mm-hmm. I have two questions regarding populations. There is one comment from Mario Cascio that about the Astra-Oxford vaccination, that there seems to be little data on older population, people older than 55. And there is another question, if there is any data available at the moment on vaccination in pregnant women. I'm not sure who might respond to that best.
So there was a EMA, European Medicines Agency, open webinar that's still online, stays online with some very good talks. And from the regulatory perspective in Europe, they're looking for the phase 3 studies to enroll at least 25% of participants to be older than 55.
Okay.
And there is some data published not from the efficacy trial, but from earlier studies of the AstraZeneca vaccine in people over 55, and it did appear to induce good immune responses in that population. And I think there'll be more detail on the efficacy forthcoming. There have been some women in the Pfizer and I think also the Moderna trials who became pregnant or were unaware that they were pregnant at the time of enrollment. The data hasn't been presented on them yet, but as far as I know, there were no safety issues. And so I think the initial recommendations, at least in the US, are that, absent any other kind of contraindication, that the vaccines could be considered for that population.
So up to now, the safety data have been incredibly impressive. The reactions have been, mild tiredness, mild injection reactions, and possibly that the response you get to the second vaccine is a little bit worse. So you might get a little bit more tired or similar to the flu sort of vaccine. There hasn't been any signal of anything more serious. When anything has been concerned, if something unexplained happened, these studies actually paused and put everything on hold. And in all cases where they were put on hold, they weren't found to be, the serious problems weren't found to be linked to the vaccines. And so the safety data so far, and nothing is ever as good as the day it's first approved. It always goes downhill a bit from there. So you should expect to hear some safety problems as we roll out access. But the safety data so far has been really impressive. and also the subgroup analysis, these big 70 or 80 page documents that are on the FDA website for the Pfizer vaccine and the Moderna vaccine seem to be reporting no big differences in subgroups. So by age or gender or by ethnicity, there seems to be pretty consistent responses throughout. And then in the background, if people were interested, these are the slides that didn't show earlier, and this is the one that sort of highlights the advanced purchases in relation to population. So anyone below the red line, any country below the red line, tells you the percentage of their population that they've been able to get vaccines already purchased for, and above the red line shows just how much excessive vaccines are already purchased by some countries. So the excess countries should be made available immediately to other lower income countries.
Okay. There is one more question. It's specifically about the Oxford vaccine and as Oxford Astra, about the knowledge of how it might affect people or persons living with chronic diseases on the long term. That I understand the data is still not available at the moment, so that would have to be analyzed in longer ongoing studies. But would you have any expectations on that?
Yeah, I think maybe one thing to clarify there is just that the weakened virus that's in the Oxford vaccine is not a weakened version of the coronavirus, of the SARS-CoV-2 virus. It's a weakened, different virus called adenovirus, which causes colds, and which, you know, most people get an adenovirus every couple of years probably, and it just causes a cold. And the Oxford vaccine is using that adenovirus as a sort of carrier for just one component of the coronavirus vaccine, which is the spike protein. It's not the entire virus, and it can't do anything bad. And so I think there's not an expectation that it would be sort of less safe than any of the other approaches.
Mm-hmm. There is one question from Robert James. I read it now. Will Pfizer and Moderna do a follow-up study on asymptomatic infections post-vaccination? Reducing illness in those vaccinated is good, but puts those unvaccinated at risk of onward infection if those vaccinated can have asymptomatic infection and the virus remains endemic.
So the Moderna submission to the FDA does include a tiny little bit of data on asymptomatic infection. It tested for coronavirus genetic material in people's nasal swabs starting after the first dose of the vaccine. And people that had received the vaccine were about 2/3 less likely to have detectable coronavirus genetic material indicative of infection in their nasal swabs after that first dose compared to placebo recipients. But I think the total number of people involved in that little study was about maybe 40 people. So they have to do more work on that. But it did provide a hint that maybe that there is going to be an effect on asymptomatic infection, which would obviously be great.
And to be fair, the Oxford vaccine is only doing this I think in a subgroup of these phase 3 studies. It's not the whole study. And if you think about what's involved from a research perspective, it's easily doable, but it does make the study more difficult. So rather than just have people arrive to the clinic twice and then go off and lead your life and see what happens, what you need to do is you need to keep returning to have maybe a swab every week to see whether there's some infections that are asymptomatic. And so it's a different approach. And you know, you could arguably say that the transmission aspect is maybe irrelevant. in the context of universal access to the vaccine.
And we have
because you wouldn't have anybody becoming seriously ill.
Okay, and we have a last question. It's from Dimitri, and I would formulate it myself with my own words. So what I understand is that his question is about if you need two doses. Is there actually does it make sense to have the two doses from different vaccines, or do they have to be from the same product or approach? And then the last question, anything about medications for treating COVID-19? So when someone fell ill, is there, yeah.
So the first question about whether you can mix and match the vaccines, I think it's in Richard's answer as well, but just discussions here is that it seems fair. Most researchers seem to think this is very easy and very likely to be positive. You'll only get this in, we have to follow the data, we have to try it and see whether it works or not, and also look at antibody responses that are produced. But most of the researchers here seem very comfortable with the idea you might get one vaccine as initial and then a second booster, especially if you're using adenoviruses. And that there's a few people I know that are already enrolled in some of these studies here, and the researchers are even encouraging them to say, if you also get access to the Pfizer vaccine, you should take that as well.
Okay.
So I'm interested, what do you think, Richard?
Yeah, I mean, I think theoretically it shouldn't be an issue, but strictly speaking, I think initially at least people are going to want to try to stick to the regimens as they were studied in the clinical trials. And then perhaps soon we'll see information from the clinical trials on exactly which immune responses will link to protection. And then that will make it easier because you can look at a vaccine and see whether it induces the protective immune response, whether it's a regimen that just includes one or two different vaccines. You'll know which response you need to get in order to see protection.
Mm-hmm. Mm-hmm.
So, and your second question about treatments, we could do this again for treatments, but it wouldn't be nearly as positive. So unfortunately, nearly everything that's been tried for treatment has failed pretty really disappointing results, apart from a UK study that used dexamethasone, which is a steroid, in very late infection. So you had to be intubated. You had to have mechanical oxygen in order for this to have a benefit, and then it definitely improves your survival responses. And then approved, there's remdesivir was approved as an antiviral for COVID-19, and its benefits are that some of the studies showed that the basis for the approval was that you recovered a little bit earlier. You recovered maybe 4 or 5 days earlier than if you didn't have access to remdesivir. So practically everything else, there's lots of hope that many different approaches will help, particularly looking at antibodies, looking at monoclonal antibodies. But lots of the large studies, not hydroxychloroquine, no difference at all. Kaletra, no difference at all. Lots of the recovery study just reported yesterday, terrible results. 1,500 people died. Half of them, a third of them were in the active arm, so-called active arm, using an antibiotic. We need different ways of evaluating treatment studies that tell us much quicker whether something's got a signal of working or not. And we could spend another hour talking about treatments and research that's ongoing, which hopefully will be more positive next year. But this year, unfortunately, didn't produce huge results that were beneficial.
Mm-hmm. Mm-hmm.
And having said that, if you are hospitalized now, the health teams, the health workers that became experienced during this terrible epidemic are much better at management. So the management is already better, much better, but it's not because of having better access to treatments, it's because of understanding better how to use ventilation and mechanical oxygen and things like that.
Mm-hmm. Okay, thank you. So from what I understand, we have included all the questions. More questions.
Yeah. I think some people posted some before, which I don't have access to them here. Was there any we missed from that?
We have included them. So
fantastic. That's great. And thanks for all your help as well for this. It's really appreciated to have so many people.
I think I've not overseen any of these questions. And today in the preparation, I've looked at the HIV iBase webpage, and there it's saying that the iBase is committed to provide a timely and up-to-date information. Having said that, I think everything around COVID-19 vaccination and treatment might or is changing quite quickly. And we might have an update webinar in the first months of the next year with more detailed and up-to-date information, and maybe also more on treatment. I must say it was very informative. I've learned a lot. I really tried hard to be prepared and did a lot of reading the past days, but still, nevertheless, I've learned a lot today. And I really would like to thank you, Simon, and also Richard for joining. And I'm quite sure that we might have another webinar next year with more up-to-date information then.
So fantastic. The website here has got there's a bulletin. iBase produce a bulletin at least every month, which includes all the updated news on treatments for COVID-19. And so there was one went online just a couple of days ago, which includes some of this information. Okay. And it's easy to subscribe online. Everything's free.
Okay. Thank you very much. And thank you to all the participants. We had 50 participants joining in, and have a good afternoon. Okay, bye-bye.
Thanks, everyone.
Thanks all.